Now we want to talk about Chapter 50 on sensory and motor mechanisms. And so the nervous system is involved with sensing information from our external and internal environments, processing that information, and carrying out some kind of response. With our senses, we have sometimes neurons themselves that are responsible for sensing information and then sending it along into the central nervous system, and sometimes we have particular cells or organs that are responsible for sensing information, passing that information onto neurons that then send it into the central nervous system. We'll see examples of both of these. Transduction is basically the modification of that signal as it goes to the central nervous system, in particular, obviously, modification from, say, a touch into a nerve impulse into something you feel inside you. And the transmission is the movement of that nerve impulse from, again, sensory neurons to those interneurons and the CNS, and then out through motor neurons. Stimuli can be of varying intensities, and that can vary based on the frequency at which a neuron is being stimulated, as we see in this one. More frequent stimulation can lead to greater intensity of the impulse. It can also be modified by how many neurons are being activated. So with touch, for example, a gentle push against you will only stimulate some of these neurons, where a more intense pressure against you it will stimulate more neurons, and so you sense that as essentially stronger pressure. Interesting line from the text. Perceptions, we perceive color, smells, sounds, taste, and these are constructions formed in the brain and do not exist outside it. This gets at the notion of if a tree falls in the woods and no one's there to hear it, does it make a sound? One answer is no, it doesn't, because in order for there to be a sound, there has to be some sensory organ there to perceive that sound. Otherwise, it's just waves moving through the air with nothing there to sense them. So indeed, this is how we perceive the world, and this information is processed in our brain. Okay, now amplification, that happens during transduction when a signal is strengthened within the nervous system. Adaptation, this is, we've all experienced this. It's when there is a stimulus that at first you perceive it, you sense it, but if it's something that becomes irrelevant, you, at least at times, can no longer sense it. So a sound that at first you notice, but if it's persistent enough, your nervous system just kind of ignores it, and you no longer hear that sound because it's not deemed as important. Alright, so there's lots of different types of receptors. Mechanoreceptors in your skin, that again, as we saw, when something pushes against you, you can feel that. Some of these are closer to the surface, which sense lighter touches, and some are deeper down for more intense pressure. There are also nerves connected to your hairs, so when your hairs move, you can feel that as well. Chemoreceptors, we have these in our mouth and nose. Certain compounds, chemicals in the air that get into our mouth or nasal passages, we can taste these things. Curiously enough, a moth has theirs on their antenna. Doesn't really matter where they are, they can still do the same job. There are receptors for temperature, and some organisms have receptors for sort of magnetic fields. It's thought that they help them with navigation. Receptors that sense heat, in particular, are known as thermoreceptors, and we have these in our skin and inside our body as well. That information is passed on to our hypothalamus, which is essentially our internal thermostat, which then can carry out some motor response of sweating, controlling blood flow, and etc. Pain receptors, we have these in our skin as well, obviously helping us to sense when there's something wrong with us. Alright, so how do we hear things? How do we maintain equilibrium? Well, in some organisms they have these structures with these things called statoliths in them, and there's all these hairs around here. And the statoliths are kind of heavy, and so regardless of the orientation of the organism, they will always be going down with the force of gravity. So even if this structure were to rotate around, say 90 degrees, the statoliths would then be over in a different part of it and stimulating those hairs. And this is what allows the organism to tell up from down. On insects as well, curiously enough, they hear from a membrane. We have a tympanic membrane in us, in our inner ear, but they can hear via a tympanic membrane on their foreleg. So here's our ear. We have the outer ear, middle ear, and inner ear. The outer ear consisting of the pinna, that cartilaginous structure that's on the outside, sort of catching the sound waves, traveling down the auditory canal. Those sound waves bounce up against the tympanic membrane, and or otherwise known as our eardrum, and it starts to vibrate, pushing against these bones in the middle ear, the malleus, incus, and stapes. And so then the stapes basically pushes against the cochlea, and there's liquid in the cochlea that then starts to vibrate, and that is what gets transduced into an electric impulse in the cochlea. So, focusing on the cochlea, I'll come back to that in just a second. So here the cochlea is this long tube that, as you can see, is kind of curled up, and this image down here is just stretching it out, and there's sort of a, so here's the stapes, and it presses against what's called the oval window and gets the signal, these waves moving through this liquid, first inside the vestibular canal and then around through the tympanic canal. And there's this membrane on the inside of the cochlea, which we're looking at here. Here's a cross-section of the cochlea, the vestibular canal and tympanic canal. So the sound wave travels down through these canals, through the liquid in this canal, and it causes this structure called the tectorial membrane to essentially rub up against these hair cells that are embedded in this basilar membrane. And these hair cells have nerves attached to them, and so when the hairs are being moved one way or the other, it changes the amount of neurotransmitters that these hair cells releasing and the number of nerve impulses that are being sent through the nerve. And so when the hairs are not moving at all, you can see there's sort of just a certain baseline amount of the neurotransmitter and a certain frequency of the action potentials or the nerve impulse being sent, but then they're bent one way and the process speeds up, bent the other way and it slows down. And so depending on the frequency of the signal being sent, we essentially perceive that as different kinds of sounds. So the cochlea, you can see again, it's curled up, so it's kind of long, and this membrane extends throughout it, and the membrane has different thicknesses on it depending on where you are inside the cochlea. And so depending on the frequency of the sound wave or the wave moving through the liquid, different parts of that membrane will begin to vibrate, and thus different portions of the different patches of those hair cells can be stimulated. So these down here and here aren't being stimulated, but the ones in the middle are being stimulated, and so we can perceive these as different frequencies of sound. And so at a point B travels or is stimulated at a frequency of about 1,000 hertz, whereas A, it takes about 6,000 hertz to stimulate it, and then further along at point C, it takes only about 100 hertz. So again, depending on the frequency of those waves, different parts of the cochlea will be stimulated. Now, you can see off to the side of the cochlea we have these things called the semicircular canals, and these are oriented in the XYZ plane, if you will, and inside of them they have this structure that has more of these hair cells on it, and these hair cells are embedded in this material called the perilymph. It's sort of a consistency of jello, you might say, and when we move one way or the other or rotate our head, that perilymph kind of moves with us, and it causes the hair cells to bend one way or the other. And this is how we can tell which way we're moving. It's also how we can tell up from down, because there are these things called ostoliths inside the perilymph that are similar to the statoliths we see up here. And so they also will be on the lower side of the perilymph with gravity, unless you get off to the side, in which case they'll move. And so that's how we tell up from down. This system can sort of be fooled or get out of whack when people travel up to the space station getting zero gravity, and in some folks it's been known to cause a certain amount of discomfort or nausea even when they no longer have gravity around. Fish have an interesting sensory system, what's called a lateral line system, and so this line runs along the length of their body, and here we see sort of a cutaway, and so down inside they have these structures that should sort of look similar to what we just saw with the perilymph. This thing's called the cupula, and so it has hairs embedded in it, and as waves move through the water, it causes the cupula to move and the hairs to move, and so fish can sense subtle movements in the water around them, and it's a mechanism they use to keep track of what's happening around them, particularly behind them, and they use it to escape from predators largely. Alright, section 3, vision. So the degree to which organisms can see varies a great deal, from relatively simple organisms like the planaria that has these pigmented eyes, in quotes, and they essentially can sense light from dark, and they have nerves attached to these cells in this eye of theirs, and so depending on how much light is being perceived, different frequency of impulses will be sent, and it allows the organism to respond in a simple way to move away or towards light or dark areas. Insects, it gets a little more complicated. They have the compound eye, and so it has many facets, each with its own lens, and cells that are able to receive those that information, that light information. And then we move up to the human eye, which gets quite complicated. And I'm going to stop the video here, and we'll pick it up from here in the next one.